Hello students, today's video lecture is on the structure of the cochlea. We have already learned the basic structure of the ear, and so let's take a couple minutes and review the basic structures before we proceed. So I have the full ear out here. Remember, the outer ear is called the pinna or the auricle. The external auditory canal leads to the eardrum or tympanic membrane. And this area that it's circled here is in zoom. So I'll get you started with letter E, that's the tympanic membrane. Go ahead and use the other choices to complete this diagram, and then we'll check your answers when you're done. Okay, let's go ahead and go over the rest of the answers. We found the eardrum. On the other side of that, we know this is the middle ear area, and so it is an air-filled cavity called the tympanic cavity. That's all this sort of lined space here. And then we have our three bones within the tympanic cavity called the malleus. Letter B is the incus. We're going just based on position, not really shape because it's hard to tell. So malleus, incus, and this one that looks like a little horse's stirrup, letter C, is the stapes. Remember, the common names are hammer, anvil, and stirrup for malleus, incus, and stapes, and those collectively make up the auditory ossicles of the middle ear. Finally, the last part of the middle ear is this tube. We know that leads to the throat, and that's called the Eustachian tube. That's how we equalize the pressure of the air in our middle ear compared to the outside pressure. As we progress towards the inner ear, we see this snail shell-like structure here. That's the cochlea. Very easy to recognize. That's where our concentration will lie today. This main area here is called the vestibule. That's letter I. And that's sort of the connector between the cochlea, which is the snail, and these three parts here, which have to do with balance. Those are called the semicircular canals. So the vestibule sort of sits between them. And then finally on the vestibule, we have two membrane-covered openings. Letter H is always going to be lower, and it should be a perfect circle because it's called the round window. The membrane-covered opening, we'll talk more about that today. And then right behind the stapes, you see this other structure, letter D. If I could remove the stapes, we would see that there's a second window like this round window, but it's oval in shape, and so it's known as the oval window. I'm going to color the oval window and the round window two different colors, and I'm going to be very consistent with those colors throughout the lecture so we can kind of keep track. Because what we're going to do is we're going to look inside the cochlea, and it's all spiraled up here. We're going to unwind the cochlea and see what's going on inside. So it will really help us if we use the same colors throughout the lecture for the oval window and for the round window. Okay, I have colored both my oval and round windows. I've chosen green for my oval window. I think that's a great choice because that's the beginning of our story. And you'll notice it's that area just surrounding the stapes. So if I took the stapes off completely and we just looked at the oval window, it would look just like you're suspecting, like this. I have chosen red for the round window because it's the end of our story. So let's go ahead and use these colors consistently throughout the lecture. Now, what we're doing is we're going to look at the cochlea inside. So this is kind of got a bony shell, almost like a seashell. And we're going to be taking a look at what the cochlea looks like inside and what's in there, because that's where our hearing happens. So let's go ahead and do some notes, and along the way, I will add some things to our diagram. So the cochlea is made of three stacked canals. And if we were to take a cross-section through the cochlea right about here, what we would see are three canals. This is just a replica stacked on top of each other, where this is an empty space, this is an empty space, and this is an empty space. And that would be right in here. I can't get the angle quite right, but those canals will reach the entire length of the cochlea and then turn around and come back. It's pretty confusing if you look at it when it's all wound up, which is why we'll unwind it in just a little bit. Anyway, there's three stacked canals in the cochlea, and the first canal will start at the oval window and then proceed through the cochlea. So because the oval window is actually on the vestibule, that first canal is called the vestibular canal. So it begins at the oval window and then reaches all the way through the entire cochlea until we get to the center of the spiral. And then it turns around on top of itself, almost like you had to do a U-turn, and comes all the way back. And this time it's going to end at the round window. And that is called the Tympanic Canal. So technically the vestibular canal and the tympanic canal are the same canal. It's just that one starts at the oval window and goes to the center, and the other starts at the center and ends at the round window. So I'll show you what's going on down here in our simplified drawing, but let me explain what we're looking at here. So here's sort of a image that allows us to see inside this cochlea. And if we zoom in, I'm going to use my colors again to represent my oval window and my round window. And we can see here that there is a canal that starts right here and goes this way. And now I'm not able to follow it really well all the way because it kind of turns back on itself, but it will reach all the way to the center and then come back and end at the round window. So again, that's still fairly confusing. So if we look at this simplified drawing, we need to get our bearings first. Here's the oval window and the round window. And if we can take this cochlea and unwind it, stretch it out, we get a structure like this where we see this canal that starts at the oval window, does a U-turn, and ends at the round window. So this first area is the vestibular canal. And as soon as we cross the center line at the center of the cochlear spiral, as soon as it starts to head back the direction it came, we change its name to the tympanic canal. Keep in mind, it really is just heading in a different direction. That leaves us with the third canal. And where is that? It's situated between the other two. So remember I said how the canals are stacked one on top of another, and there's three. So we've got the vestibular canal on the top, the tympanic canal on the bottom of our unwound cochlea, and then we've got this space in the middle. That's the third canal. And that canal is called the Cochlear Canal. So to keep things straight, I'm going to use some colors to represent each one. I'm going to color both the vestibular canal and the tympanic canal this blue color. And I'm choosing blue because they are filled with a fluid. That's a major part of the story, is that they're filled with this watery fluid. So I'm going to color both vestibular and tympanic canals blue. So I've colored both the vestibular and the tympanic canals blue. I am going to do something else. May seem a little extra, but I'm going to make little waves in that fluid so we know it's a fluid. The third canal, that cochlear canal, the space in between, I'm going to be very careful and color just the background area within that space because I've got some hair cells here that will have their own color. So I'm just going to color this background area here for the cochlear canal, and I'm going to choose a really light color, visible, but not really super obvious because I'll just color over some of the areas. Okay, I've colored my cochlear canal space in this sort of light yellow and made a note up here that that's what that is so we know. So we've got our vestibular canal, our cochlear canal, and our tympanic canal. This is all set up for what's inside of that cochlear canal. So what we're looking at is a very important structure that is directly related to our ability to hear, called the organ of Corti. And I'm going to make a bracket here to include the structures of the organ of Corti. Very specific. And it's got this membrane at the top, membrane at the bottom, and the hair cells in between. Within the cochlear canal lies the organ of Corti. And the organ of Corti is made of three main structures. It's like a sandwich, really. Membrane, hair cell, membrane. So the upper membrane is called the tectorial membrane. And I remember that it's upper because tectorial, T for top. The bottom membrane underneath is called the basilar membrane, and I remember that B is for bottom. And then stretched between the two are the hair cells. And I'm going to go ahead and color a couple of the hair cells in just a minute so you can understand what they are. Remember, those hair cells are not real hair. They're just cells with really long cilia. And the cilia, as you can see, is embedded into the tectorial membrane. Okay, now for some colors. Okay, so I've done quite a bit of coloring. My tectorial membrane I did in this darker purple. Remember, that's the membrane that's on the top of this organ of Corti. Basilar membrane on the bottom is a lighter pink, so that's going to be here. And then the hair cells are stretched between those two, and I did a few of them in orange. What I've also done is draw the organ of Corti by itself down here. And number 1 is the tectorial membrane, number 2, the bottom basilar membrane, and 3 hair cells. So the difference between this and this is the cochlear canal in the background. So if I removed this structure from here, all I'd have is this yellowish space between the other two canals. All right, we're almost there. So how does this all work? Let's take a little bit more realistic look at the different canals of the cochlea on the next page. Here is the semicircular canals and vestibule and cochlea. Let's get our bearings with our oval and round windows. My oval window will be here and round down here. So we know that this is the cochlea here. And notice they've taken a cut through it, and we're looking at that here. So we have canal number 1. This space would be the vestibular canal, the tympanic canal, and the very light colored cochlear canal with the organ of Corti that I kind of just colored over. So if we zoom in on that cochlear canal, here's our organ of Corti. And I know it looks a little different, but if I use the same colors from before, I think you'll get it. Here's one of the parts. It's not quite as sandwich looking as my unwound cochlea drawing up there in the last picture. Here's a second part, and the third part is here. So just like on the other page where we had purple, orange, and pink in our organ of Corti, we have the same more realistic drawing of it here. Let's go ahead and label that again. Another view of the organ of Corti and its 3 basic structures. The purple is the tectorial membrane, T for top. The pink is the basilar membrane, B for bottom. And the hair cells are between them. Remember, not real hair. And the cilia of the hair cells, we can see right here, is embedded into the tectorial membrane. So collectively, these 3 parts, tectorial membrane, hair cells, basilar membrane, make up the organ of Corti. And another thing that we have in this image that I feel is noteworthy is the nerve endings that are coming off of those hair cells. Because remember, ultimately, we're trying to figure out how hearing works. And these hair cells are really important in that process. So they're actually connected to specialized neurons. And all of these specialized neurons will collect to form a nerve. That nerve is visible here on our first page. Part of that nerve is coming off of the vestibule and semicircular canals to help us monitor balance. That's a different story. And part of it is coming right out of the back of the cochlea. And that's for our hearing. So these hair cells that are embedded within the cochlea are going to stimulate the neurons in that nerve. All right, let's put all the pieces together. As we are hearing, sound waves are collected and funneled by the very outermost part on the outside of your head called the pinna, or sometimes called the auricle. Those sound waves will travel down the external auditory canal and cause the tympanic membrane or eardrum to vibrate. So sound waves funneled by the pinna, that's the outer part, collected and sent by the external auditory canal to our tympanic membrane here. On the other side of the tympanic membrane are the three tiny bones, the malleus, the incus, and the stapes. So as the tympanic membrane vibrates, we can see the malleus is actually connected to it. Here, at the handle part, and so it causes the malleus to vibrate, which causes the incus to vibrate, which causes the stapes to vibrate. So we're just going to pass vibrations right through all three of these auditory ossicles. So malleus, then incus, then stapes. Now, what is the stapes attached to? It's difficult to see in this app, but right behind the stapes, let me change pictures. Right behind that stapes is the oval window. And we know the oval window is actually the beginning of our vestibular canal that's filled with fluid. So if this stapes is vibrating back and forth and this oval window is a membrane-covered opening and that also vibrates, that's going to create waves in the fluid of the vestibular canal. And those waves are going to travel all the way around this spiral to the middle, and they're going to continue on through the tympanic canal until they get to the round window. And then the round window is going to act like a dampener. It's going to absorb the pressure of those waves. So what is the purpose behind all of these structures in the cochlea? It all comes down to when these waves in the fluid of the vestibular canal pass by, they disrupt this tectorial membrane. And the tectorial membrane starts to wiggle, which disrupts the cilia. And if we look at our other diagram at the bottom of the hair cell, there's nerve endings. So the tectorial membrane vibrates in response to the fluid, which wiggles the cilia, which stimulates the nerve, which sends electrical impulses about what's happening to our brain. Let's fill in the rest of those notes. So the vibrations of the auditory ossicles, Malleus, incus, stapes, are passed onto the oval window located on the vestibule. As the oval window vibrates, the fluid in the vestibular canal will also begin to vibrate, or what we'll see waves, like waves in the ocean. And as soon as we have waves in this vestibular canal, the tectorial membrane will start to move. Okay, it's like if you had a raft on the ocean and big waves come by, you're not staying in the same place, you're also going to move. So the upper membrane, the tectorial membrane of the organ of Corti, is moved in response to these waves. Movement of the tectorial membrane causes the hair cells to move. And as they do, they generate electrical impulses. Here's the main goal. The hair cells generate electrical impulses. These electrical impulses travel out the neurons attached to the hair cells and to the auditory nerve of the brain for processing. So that auditory nerve actually starts here at the hair cells and collects to look more like this. And that's cranial nerve number 8 out of 12. There's 2 branches. There's a vestibular branch and a cochlear branch, but collectively we call it auditory nerve, or sometimes the vestibulo-cochlear nerve. So that's the process of hearing. I also wanted to talk to you quickly about how balance works, because originally we said the ear is responsible for both hearing and balance, like you feeling like you're balanced and not tipping over or dizzy. So let's talk quickly about how balance works. It works very closely to the way a snow globe works. So we know the balance happens in the semicircular canals. Remember, that's this section here. And the 3 semicircular canals, remember 3, 1 for each of our 3 dimensions, they are also filled with fluid. This fluid moves when we move. And the semicircular canals also have hair cells in them. And these hair cells are positioned at the base of each semicircular canal. So if you look down at the bottom, you can see that there's kind of a larger part at the end of each semicircular canal. So they're in this like little swollen area here. The long cilia of the hair cells are embedded in a gelatinous material. Here's where it sounds like I'm making this stuff up, but I'm not, I promise. Think about like a Jello mold. People used to make Jello molds back in like the 70s and 80s. So the hair cells are embedded in this gelatinous material, and then on top of the gelatinous material, there's a little grouping of tiny crystals called otoliths. That literally translates into ear rocks. So if you've ever heard someone say you have crystals inside your ear, they were not lying. They're little tiny crystals, and they're situated on top of the Jello mold, and there's hair cells in the Jello mold. Let's look at the image. So if we were to look here, or here, or here, or here, or here, what we would find are, we'll use our same color for hair cells. Here's the hair cells, and here's the cilia. Okay, I'm just going to do one. And I'll mark my color up here. And then you can see that these hair cells are embedded in the gelatinous material. So I'm going to pick a Jello color. Let's go with lime Jello. Maybe this. Here's the gelatinous material in green. And then what color should the otoliths be? I don't know. If they're crystals, maybe a gray? Here's our crystals at the top, like little grains of salt. I'll mark my otoliths as colored gray. All right, so what is this all about? So the way we know that we're in balance or unbalanced is really by movement of our head, because that's where the mechanism is that notices. So when we move our head, if we tip our head to the right or we tip our head to the left, that's going to move these crystals, which is also going to shift this gelatinous material and the hair cells' cilia, because we've shifted. Now gravity is taking a hold of these parts. So motion of the head moves the otoliths. This shifts the cilia of the hair cells embedded in the gelatinous material. Okay, so it's like we're tipping our Jello mold to the side. Of course it's going to move. Moving hair cells stimulate the nearby neurons. That's these little strands down here. We'll color those yellow. So moving hair cells stimulate the nearby sensory neurons, which send electrical impulses out the auditory nerve to the brain. So this little Jello mold with the crystals on top of it and the hair cells embedded in it is all positioned at these red circles. And if we look at where the nerve is positioned in this one diagram up here, this is the nerve section for balance, and this is the nerve section for hearing. And then, of course, we would call that the auditory nerve or vestibulocochlear nerve. That's cranial nerve number 8. So there you have it. The structure of the cochlea, the process of hearing, and balance.